So we are going to start about the DNA sequencing and the data analysis part. So we'll cover today is the basic concepts and some of the analysis about history that before we have next generation sequencing technology, and also the experimental considerations.
Is it this thing doesn't work? Stop. Why this doesn't move?
Okay. All right. Just recap what we have talked about before I left. And we discussed about the file formats. That's a big topic in the next generation sequencing data analysis. And these are really not the gene list that people present to you after their analysis. This is a different level of the, if you will, the raw data that we have to deal with if we want to get into more creative about how to deal with the data. So we started from the FASTQ files, which give you the raw sequencing information. And also we talk about the BAM and SAM files. Those give us the alignment information. And in this BAM and SAM files, there are many different fields, and there are many different type of tags. And we went into very detail about how to interpret individual ones. But I have to mention that even we gave a pretty thorough interpretation on that. It's still only a part of the story. And those files are constantly evolving as the experimental procedure on next generation sequencing become more and more complex. So you will always need to go to the individual file menus to deal with the individual details. And then the last standard file formats we discussed is called VCF files. This tells us where individual variants are. And those are, so for the VCF files, normally for each individual file that can contain either one patient or up to 10,000 patients or even more. So as I mentioned earlier that in the 1,000 Genomes Project, there is only one VCF files being distributed, that all the 2,500 individual, their genomic variation are in that particular file. And both these two-level files can be indexed and can be queried in a very, if your data is stored in the right format. And we also explained a few variations of other formats, including the BAT files and the Vigo files, and there's several variations of those. And those are the files that, in many cases, are the ones that we need to deal with for the data analysis. So when we go into the situation of the data analysis on, for example, ATAC-seq or ChIP-seq, so we will pretty much use the BAT files and the Vigo files. So, but just stay tuned on that. 2 weeks ago, and Guanglong gave a guest lecture on this. So basically, that is about a few different type of visualizations on the next generation sequencing data. And the two include UCSC Genome Browser and the WashU Epigenome Browser and the Integrative Genomic Viewer, and also circular plot. Those are the softwares that are very, very critical for us to deal with the data analysis. And especially when you try to have some creative ideas how to deal with the data analysis. And in many cases, these are the softwares that even myself personally use, not only my students, but myself, and use quite a lot about these different type of softwares. And I would love to staring at the browser to see how the sequencing rates is stacking around. And so some of the ideas will come along with it. And there are many different ways that to load the data. And I think Guanglong gave, I went through the video a little bit. Guanglong gave a very good, thorough interaction. And if you have, interpretation, if you have any specific question about that, you can either ask Guanglong or maybe talk to Jill and or anybody in my lab, but they are pretty much familiar with this. And last week, Ed gave a guest lecture on the high throughput performance computing system at IU Medical School, and this is the system that we are using to really do the calculation. And so basically, there are different computational nodes, including the CAST, Carbonate, and the Big Red tool. Again, these are just the basic concepts, and from time to time, some of the systems is going to retire, and new system will come in. But in general, the overall structure remains the same. And so those are the computational nodes, so that where your job is being submitted, and your data is actually being put into a separate file system, and it's either DC1 or DC2 system, and all the data will also be backed in the SDA system, which is a tape-based scholarly data archiving system. And what I heard about is the UITS is in the process of upgrading the DC2 system into the DC2 Plus, which have faster access as well as bigger space. But again, the structure will remain the same. It's just the specs and the command to access these servers will be slightly different. Okay? Any questions so far? Okay, let's move on. In the next couple of lectures, we will focus on the DNA sequencing analysis. So today we'll talk about the basic concepts, history before the NGS technology, and experimental considerations. And the next lecture, we'll talk about some of the bioinformatics components of the DNA sequencing analysis, including the mapability considerations, refined sequence alignment, and the quality recalibration. And those are part of the GATK package that when we do the DNA sequencing data analysis, that those are the ones that are being used very, very frequently. And then after doing that, we will talk about the variant identification, identify individual variants, and structure variation identification, and copy number variation identification. So those are very important topics as well. And so this is, some of them are also our standard data analysis pipelines. And after doing that, we'll talk about, we'll have a guest lecture from Dr. Dongbin Lai, talk about statistical genetics and its applications in the NGS data analysis. I specifically asked him to give us a little bit more introduction on the data analysis even before we have next generation sequencing, such as GWAS analysis, linkage analysis, and then the current next generation sequencing based analysis. It should be very, very informative. And then we'll come back to me October 1st. We'll talk about variant prioritization. So basically, once we identify the individual variant, how do we rank which one is more important? And because a lot of them are not very important, they're called passenger mutations. So we'll discuss about that topic and using one full lecture. And then we'll have a guest lecture from Dr. Matteo Vata, and who is an adjunct faculty in our department. And he will talk about the regulatory and ethics considerations in the clinical sequencing. And then towards the end in this lecture series, we'll talk about high throughput reporter assays. And this is following up with variant prioritization. I would consider this as a variant screening part. So it will be very, very, this is another lecture that we added in this year. Okay? So that is the plan. And after that, we're not too far away from the midterm exam. I'm pretty sure everybody is ready for that. Okay, so we'll talk about today and different type of genetic variants, and then the state of the genetics of the complex disease before we have the next generation sequencing technology, and some of the applications But I will focus. I will spend some time talking about experimental considerations on the DNA sequencing, which it will include whole genome sequencing, target sequencing, pooled sequencing, long-range genomic sequencing, as well as single-cell genome sequencing. Okay. The first part is what are the genetic variations? Just to give some of the brief concepts. And there's multiple types. The first type that most people are talking about is called SNP. It's a single nucleotide polymorphism, which is meaning it's very straightforward. Only one nucleotide is going to be different between your DNA sequence comparing to the reference DNA sequence. And normally, we will have one SNP in every few hundred bases, sometimes in the 1,000 base level. The second type of variation is called short indels or insertion deletions. And as the name implies, so you can see that this is not one base difference, rather it's a few of the nucleotides are being inserted or deleted from the reference genome. So you can see the total number of the nucleotide changed. So this type of variance happens one in every few 1,000 bases. And then the next type is called a microsatellite repeat. And the number, this type is the third type of genetic variance. It happens one in every few 1,000 bases. And the definition for that is 2 to 6 base repeat units. So you can see this one is a TCA, 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 TCA. In reference genome, probably there are 4 copies, but in the donor genome, there's only 2 copies. So the numbers of this repeat differs, and that is called microsatellites. Making it bigger, there is another thing called mini satellites, which are one in every few 1,000 bases, and this is the same as microsatellites, except for the repeat units become bigger, and they can be from 10 to 100 bases. And those are called mini satellites. There's no clear differences between microsatellites and mini satellites, except for it's just the length of the repeat units is different. And then there are further some type of variants called repeated genes, and some of the genes occurred multiple times in the genome. So it's just again that units of the repeats is becoming bigger. So those are called repeated genes. And in addition to all this, there is another type of large structure variation. The genetic variants called large structure variations, and they will include insertion deletions. Those are we normally refer as big insertion deletions. Sometimes it can be a few 1,000 bases. Sometimes it can be a few 100,000 bases. It can be very, very long, and it's a big chunk of genomic regions that are being inserted comparing to the reference genome or being deleted. And there's duplications, and some of the parts will be occur multiple times. And there's inversions, copy number variations. So all those are being defined as structure variations. So we will spend a few part of one lecture talking about the algorithms to identify the structure variations using the next generation sequencing data. So we'll come back to the SNP a little bit. In terms of the single nucleotide polymorphism, is defined as a single base change in the DNA sequence that occurs in a significant proportion of a large population. Okay, so the reason that I want to mention this is, people know what SNPs are, but sometimes we ignore one important concept here, which if you only have one nucleotide difference, and if that variant is only you have it, that is not considered as a SNP. Okay, so the SNP needs to be called a polymorphism, and in order to qualify it to be a SNP, you need to have a significant proportion of a population have it. Okay, otherwise it's a mutation. Okay, so actually it's just for you to have a general feeling of this. There's no strict definition whether it's 1% or 0.5%, but you get the point. You need to have a significant proportion of the population have it. That is considered as a SNP. And in recent days, when we read papers, people often talk about the single nucleotide variation, or SNV, and that is more frequently used than SNP now, because there's no requirement for the proportion of population in that. And for the SNP, normally it's 1 in every 300 in the human genome, and there are over 10 million SNPs across the genome. And the SNPs are conserved across genome, often in patterns called haplotype. So what this really means is if there are two SNPs, they are like a few hundred bases away from each other. They are not independent, so they often travel together. And that is, so the pattern of that is called a haplotype. So if you are not familiar with this, don't worry, we'll come back to this concept a little bit later and talk about haplotypes. And SNPs are the most simple form and the most common source of genetic polymorphism in the human genome. Over 82% of all the human DNA polymorphisms are associated with the SNPs. So those account for over 82% of the genetic variants. There are some important concepts, and I'm sure that this part will be in the exam or quiz. So basically the concept of the transition and the transversion. Okay, so the so-called transition is a substitution between purine or between permitting. So meaning A and the G or C to T. So this type of substitution is called a transition. While transversion is the substitution between a purine and a permitting. Okay, so A becomes C or T, and G becomes C or T, C becomes A or G, C becomes A or G. Okay, so this is the transition, this is the transversion. There is an important application of the transition and transversion in the next generation sequencing data analysis. So if you read this sentence, I hope this will make sense. That is the ratio of the numbers of transition to the number of transversion for a pair of sequences. It should be 0.5 if there's no bias towards either transition or transversional substitution, right? So let's say if I have an A and I have twice as a chance that it become a transversion because I have 2 more letters to be changed to compared to transition, which is only 1 letter to be changed to. Okay, so if every change is random, and actually the transition transversion ratio should be 0.5. Does that make sense? Because the transition, you have only 1 way to go. Well, for the transversion, there are 2 ways to go. However, in our genome, this is not what happened. The transition transversion ratio is around 2 to 2.1 in the whole genome, while in the exon region it's 2.8 to 3.0 in the exon regions. Okay? Yes?
So what are the factors that promote either transition or transversion?
So you're asking why this is so far away from the 0.5. Is that correct? Yeah. Can anybody have an answer for that or have a hint?
Because DNA is a chemical compound. So
DNA is a chemical compound?
and if you want to change from A to G, or at least change for the chemical compound, they will relate to the reaction of the chemical reaction.
Okay.
So
So it's it's a bigger change, and to.
I think there's some reaction energy problem for the.
Well, I don't know that part. I mean, it could be, but actually A and the G, they are more similar to A and C and T, I think. Yeah. So that's why I think A to G change is easier than A to C or A to T change. That's why that it has a higher frequency. Is that right? Yeah. I mean, that's a good hypothesis. And I think I agree with that. So some of the molecule changes is easier. There's smaller changes and than the other two. And another important factor is evolution conservation, I think. And some of these changes will cause the organism in terms of the amino acid sequences to be changed into a more distal ones, and therefore that will cause the organism to be less fit. So there are some of the evolutionary pressure on that, but at the same time, that A to G is similar and comparing to A to C and T, so that change can happen a little bit easier as well. Okay? So these are the factors that normally people don't pay too much attention to, but when we identify the variants, one of the, let's say for one particular sample, we identify 30,000 variants in the exon regions, and the translation-transversion ratio is something that I will look at because I want to see it's close to 3.0, and that means most of the identification is correct. If I'm seeing that the average translation-transversion is 0.5, I will be very much concerned. Okay? So that is actually become part of the quality control for the next-generation sequencing data analysis. Well, structure variation, so I just go through a few examples. It's traditionally defined as deletions, insertions, and inversions of larger than 1 kb, and often involves the repetitive regions of the genome and the complex rearrangement. And before we have next generation sequencing technology, there is a very, very few tools that we can use to detect the structure variation systematically. And NGS obviously changed this fact. So structure variation affects a large fraction of the genome as compared to the SNPs, rather than just only one change. But this is when we look at the Southern Genomes data, actually people identified over 1,700 structure variations that affect the coding sequence of the DNA, but those Southern Genomes data are actually healthy individuals. So if we look at these schematics, and in the reference genome, this is A, B, and C. Those are not individual nucleotides, rather it's a region in the genome. Deletion, and you can see that B is gone, and insertion, and there's one more segmentation being plugged in, and inversion here. So you can see here is A, B, C, and then inversion here becomes C, B, A. Copy number variations, so we can see multiple copies of A here, and then segmental duplication, so you can see that this A, B, C, and there's another A, B, C that is being attached. Okay, so again, the structure variation can be more complicated than this, but these are a few of those that is currently being more systematically studied. Okay, and another point I want to make here is most genetic variations have no effects. Okay, and most genetic variation in the human genome are silent variations. They have no phenotypic impact, and they do not occur in coding or regulatory regions of the genes or within these genes but have no effects. So there are a lot of variants. So if you look at two individuals comparing to their whole genome sequencing, normally over 3 million SNPs variants will be detected between two individuals. But most of them are not changing anything, not influencing anything at all. And our whole purpose here in the bioinformatics analysis is to identify those that are important. Okay. Well, so since although most of them have no obviously phenotypic impact, they are still very useful. And you can see there are many different types of applications, including forensic science and paternal test, archaeology, and the flow of the ethnic migration, and also some of them can be used as biomarkers for select patients in the clinical trial. So even though that they are not phenotypically or mechanistically changing the functions of the genes, they can still be very much useful. Okay? And we will spend a whole lecture talking about the prioritization or the selections of the functional variants that some of them are called nonsense, frame shift, non-synonymous, and the different type of variants that could impact the gene functions. Okay? Any questions so far? Okay. So in the next couple of slides, I'm going to talk about the states of the genetics of complex disease before we have the next generation sequencing technology. I don't remember whether I've shown this figure or not, but clearly that from 1950s, and if in the 1950s, if we sequence one base of nucleotide, it would take one person a year to do that. And then fast forward to 1970s, and our capacity increased dramatically. That is when the summary sequencing is being invented. And at that point, people can sequence 15,000 nucleotides in 1 year. We are talking about one person. And this one is talking about 10 years ago, so there's a lot of zeros that are being added. And so far that in our lab, that one person can sequence this many nucleotides in 3 days. So you can see these are the numbers for a year. So this is clearly, I've talked about this, our capacity of doing the sequencing increased dramatically. And so this figure has been presented multiple times, but you can see that we can sequence things very fast and much cheaper as well. Okay, so now let's talk about the analysis, the genetic analysis before we have the next generation sequencing. And in the very early days, and we're talking about the 1980s, and when people do statistical genetics, the most usable genetic marker is the microsatellites, and based on the length of the microsatellites. And there are 300 to 400 those markers across the genome. But even based on those very, very low resolution of the markers, we are still able to identify a lot of important genetic regions that are specific to certain disease. And then after that, people start to focus on the SNPs with the technology development. So initially, there are SNPs being, the chips being designed to target about 6,000 SNPs, and later become 100,000 SNPs, and then become 1 million SNPs. I think the most dense chip so far is have 5 million SNPs on those chips. That it profiles very, very high resolution of the human genetic variations. Well, in terms of the analysis, when we did not have a very dense genetic markers, and people are using family-based linkage analysis to identify the region of the genome that is associated to certain disease. And then after that, people are basically doing a lot of on the GWAS, genetic association studies. Dr. Dongbin Lai will talk about these two topics in a much greater detail in the next couple of lectures. But I will give you a prime of the GWAS study, which stands for Genome-Wide Association Studies. And they are commonly used to link genetic variation with a disease or traits. So you can see here, there are two SNPs, okay? And there are different persons. And each person has a heterozygous variance or homozygous variance on this particular SNP. And in the first study here, so you can see that there are a lot of people who are not sick. Those are the controls who are not sick. And this is the ones that are sick, which is the case. And in comparing to this, so you can see there are a lot of people, there are purple people. Purple people are the ones that has a homozygous reference, meaning they don't have the genetic variants. But there are orange and red people. Those are the people who has either heterozygous variants or homozygous variants. So you can see that in this case, just without really going into too much detail, you can see in the case group, for this particular variant, in the case group, it's much more colorful than in the control group. And based on this, you can calculate a p-value to say whether this particular SNP is going to be associated with this disease. So this is a trait that can be either a quantitative or it can be a qualitative. So basically it can be binary or it can be a continuous variable. But the point is we want to see whether the occurrence of this SNP is associated with the trait. So that is the genome-wide association studies. So if genetic variants are more frequent in person with a disease, the variants are said to be associated with the disease. So that's the definition. And you probably have seen this type of figures in multiple contexts. And this is a very nice review paper published many years ago. But what it tells us is the underlying hypothesis of the GWAS is actually common disease, common variants. Okay, so what that means is common variants present in more than 1 to 5% of the population contribute to the common disease. This is the hypothesis of the GWAS. So let me lead you through what this figure really means. X here is the allele frequency. So you can see that here are the SNPs that are more common, and here are the SNPs that is very rare. Y here is the so-called effect size. So what that means is if for the people who has the variants, comparing to people who doesn't have the variants, if the people who have the variants have like a 30 times higher risk to have that particular disease, that means that has a very large effect size, okay? So if we look at this, and some people, the low-hanging fruit will be here, okay? So those are very common variants. At the same time, the one that has it has a very, very severe impact on the phenotype. So common, at the same time have large effect size. But these variants, are either not existing or they are impact a phenotype that is not very, very important for the survival of the population, right? So like our skin color or eye color or hair colors. So those are the ones that it could be it could happen very, very common at the same time, has a larger effect size. But normally, it's not going to happen in the disease situation. That otherwise, if something that happen very common, and then if it happens, and then the people will get sick, and normally those are either Mendelian disease or it's not happening. And another type of variants are located here, which they are not very common. They are very rare. But once it happens, and the impact on the phenotype is very, very severe. So those are the ones that normally is a Mendelian disease. For those of the variants, the previous analysis, the way that people identify those are using family-based linkage analysis, which Dongbin will talk a little bit more detail in the future. And on this side, those are the ones that are common, but effect size are not very high. Okay, so which means that those SNPs occur very frequent in the population, but if you have it, well, your risk of being sick is like a 1.3 times higher than otherwise. I mean, it's not that much predictive, and but at the same time, it's each individual variance only contribute to a very, very small effect size. And in this part, normally we don't care about that. Those are not very common, and once it happens, it's not very severe on the phenotype anyways. So for the GWAS study, this is the part we are focusing on. The common variants, common disease, common variants. But if you look at this spectrum, one of the important things to notice is, by definition, they are usually a very, very small effect size. Okay, and they generally do not capture the rare variants. They only focus on this part. And one of the reasons for this is if we do the GWAS study, we have to use the SNP arrays. And then, as we talked about in the first lecture, and if the SNP array, if we put something on the SNP array, that means we know there is a genetic variance. Normally, people don't put rare variants on that because it won't profile too many people. So only common variants will be on the SNP arrays, and that's why that we are stuck in this size. With the next generation sequencing technology, if we design the study properly, and this is the whole spectrum that we will be able to identify. So that is the central point of this particular figure. And the genome-wide, again, I want to point out that the GWAS study is only focusing on here, common disease, common variants, but normally the end result is a very, very small effect size. And there are some of the successful GWAS stories. The significant association reported through March of 2010 are here. There's over 800 SNPs. But since then, this list goes bigger and bigger, and there is actually an online tool I'm listed here, and you can search different phenotypes, and then what are the SNPs that are associated with those phenotypes. So you can see all these different things are available here. Okay. So now let's talk about the limitations of the GWAS. And the disease and trait associated SNPs are not necessarily causative variants. Okay, so just because a SNP is associated with a phenotype doesn't mean this SNP caused that phenotype. Does that make sense? Okay, so the reason for that is now we're talking about this so-called LD blocks. LD stands for linkage disequilibrium. And so you can see that within this block, there are many different SNPs. But these SNPs, because of the evolution, they always come together. Evolutionally, they are conserved. So this, which means if we know this SNP, and we will know this SNP as well. Okay, if we know the genotype at this location, we will also know the genotype at this location. So everything within this are linked together. So that's why that sometimes when we identify the SNPs, that we say they are associated to the phenotype, and it could be some of the other SNPs within the same linkage block, actually they are causing the disease. So this is particular SNP is only a tagged SNP in the LD with the causative variants serve as a genetic marker for a region of interest. And identification of causative variants would require further studies, for example, resequencing the entire regions. And those are like something like this. Yes?
So what would make us SNP if not a causative, if it's not causative for the phenotype, what would cause it to be associated with the disease itself?
So let's say, for example, that's a good question. For example, this SNP is causing the phenotype, okay? And but these two SNPs, they are in the same linkage block, which means that if I know this genotype of this location, I will also know the genotype of this location, okay? So when people design the SNP arrays, they are not going to put both two SNPs on the chip, because one will be representative to the other one as well, right? So in the case that they only put these SNPs on, so this SNP is going to be associated to the phenotype, but this SNP is not causing the phenotype. The only reason it's associated with that is it's in the same LD region with the causative SNP. Does that make sense?
So the only reason would be it's linked, otherwise.
Yes, it's a tag, it's a marker. Okay, it's not causing that at all. So that's one of the reasons that now the NIH, if you go to the RFA or whatever, and one of the major interests people have is from the association to the functions. Okay, only looking at the association, we still don't know what are the causative variants that lead to the disease. We need to have some strategy to find out the causative variants. Yes?
So in this example, how are the three LD blocks related?
Well, they are not related. So this only means that this SNP is tagging this LD block, this SNP is tagging the second LD block, and this SNP is tagging the third LD block. Okay. Normally, they will choose one or two SNPs in each LD block to tag that. Yes.
Like, how do you start on if you have to identify these variations starting from the other studies? How do you start on it? Because you don't know which block is like there's a variant on which block, right? So start working on every block, non-block.
Yeah, so basically the statistical genetics part is, let's say we have 3 blocks. Each block have a tag SNP. And then what the GWAS will do is, for each individual tag SNP, they will look at the association between this SNP and the phenotype. So if we have 3 SNPs, tag SNPs, we will do this 3 times. If we have 5 million of these SNPs, we'll do this 5 million times. And that causes a whole different type of problem, which is called multiple hypothesis correction problem, that Dongbin will go into a little more detail on. But we will treat individual SNP on the array as independent and to look at the association between this and the phenotype. Does that make sense?
It does, but then like you are associating this particular SNP to the phenotype, but you don't know there's a causative variant, a different causative variant in the same block.
Yeah.
How do you start?
How do I, from this one, if I say identify this is a...
With the GWAS data, I don't know like this block is causing it, right?
That's a big question that people is trying to figure out how to answer that question. So once I say, okay, block 3 and is associated with the phenotype, what are the causative SNPs? How do I further search for the causative SNPs? And then to be associated with the phenotype. And I think I don't have a strict answer for you, but we will have 2 lectures trying to address that. One is the so-called variant prioritization, and we will look at all the variants in this LD block and to predict their function, whether it's important or not. And the other one is the high throughput reporter assays that using the experimental approach to screen which individual one can cause the molecular phenotype change. But there is no systematic way of doing that. Okay. So a lot of people mentioned that the GWAS limitation is the missing heritability, and so basically it comes from this, right? And comes from our initial figure, like the GWAS only focus on this part. It's a common variance, but the ones that you identified are normally very, very small effect size. So you can see here is the median odds ratio per copy for the risk allele is only 1.33, which means it's only a little bit increase of the disease risk compared to the ones that doesn't have that particular SNP. And we're talking about those are the ones that GWAS already found out they're associated to certain disease. Okay, and why is that? There are many different type of interpretations. What are the other factors that contribute to the disease heritability? Like there are many of the SNPs that are much less frequent, and like less than 1%, and they are not being profiled on the SNP arrays. And so I'm putting on here. And actually in the Southern Genomes Project, people identify over 11 million novel SNPs. And those are the ones that is not on the SNP arrays. And another important reason for that is there are interactions between genes and between genes and the environmental factors. So those are gene-gene interaction or gene-environmental interaction. Only based on the genetic study or GWAS studies, we are not going to identify those. And also contributions of structure variations are not being profiled in the regular GWAS studies. But there are also many other reasons, including epigenetic effects and many other variants that we don't know what their functions are, and we were not able to profile at this point. Okay, so I think I just want to give you a very quick prime on the GWAS study, which don't mean we'll talk a little bit more, but keep in mind this part. I want you to understand the hypothesis of the GWAS is common disease, common variants. Okay, and then based on that figure, very important figure, common variants indicating their effect size are normally not very big. And so those are the things about the GWAS that we need to think through and how to interpret the data. Okay, so the next section I want to talk about is to do the DNA sequencing studies. There are personal genome sequencing is the one that we have a lot of interest. I think the second part is the story of the little kid, and his disease was cured, if you will. And after the next generation sequencing intervention, we talk about it in the lecture 2, as I'm not going to talk about it again. But I want to discuss, I didn't talk about this, right? The clinical assessment of a 40-year-old man who presented with a family history of coronary artery disease and sudden death. And this is a very interesting paper that published in Lancet in 2010. And so basically this is one that is more like a perspective paper than to see that in the future, how the authors are envisioning that we are using this next generation sequencing technology in the clinical practice. This is not cure a disease. This is something that they want to use as a regular examination of our fitness. So this paper talk about a 40-year-old man, which is a professor at Stanford. And he has a family history of coronary artery disease and sudden death. And this person appears well, and the medical history was not clinically significant. The patient exercised regularly and without symptoms and taking no prescribed medication. So clinical characteristics were within the normal range. So healthy individual with some of the family history of certain disease. If you look at this, this is a report that you will get today if you go to a hospital to do a regular annual check. Okay, if it's you have a good insurance. Sometimes they only weight me and say, oh, you're okay. but sometimes they do check a little more intensive. They say here's age, weight, height, weight, body mass index, and cholesterol level, and many other things. So those are basically the clinical readout. Okay. And this is the family history of heart disease of this particular patient. And this is the patient himself, 40-year-old, and everything looks right. But if you look at all these different small squares, and those tells you that what different disease that might be. So you can see this clearly has a family history of heart-related disease. Those are heart-related disease. Okay. And what this person did is did whole genome sequencing and using a technology that is already out of the picture. It's being out-competed and it never goes into the market. And it was called helicose. So you can see that this is a very heated, competed market, I guess. And back in those days, they did 148 gigabase of the raw sequence. So what that means is if you think of human genome is 3 billion letters, 150 gig means 50x coverage for the whole genome sequencing data. And those are a strange technology which I'm not familiar with. And they are by average 33 base long reads. And they were able to detect 2.6 million SNPs and 752 copy number variations. And they did some validation on some of the clinical interpretable variants, and the copy number variations were also validated using the PCR. But those are not the major point of this particular paper. And there's a disease and risk analysis, and basically they look at it very, very intensively of how to annotate those variants, those 2.6 million variants that they have identified. They look at the variants associated with genes for Mendelian disease. There are databases about that. They talk about novel mutations. They look at the variants known to modulate response to pharmacotherapies, so pharmacogenomic studies. They look at SNPs that previously associated with complex disease. So those are all different type of databases. And they also look at the database queries about disease-specific mutation database, and or more generic ones like HGMD, which is a human gene mutation database, or OMIM, online Mendelian inheritance in men. So all those databases, very, very intensive database search, and they also look at the predictions based on allele frequency, the evolution conservation, and protein domain disruption, and so on and so forth. So those are the database or prediction tools they have used, and very, very intensive. So here is an important figure, what they get out of it after they did go through all these troubles. So let me show you what this really means. So X here is a clinical risk, okay, from 0% to 100%. And the Y here is each individual is different disorders, like obesity, coronary artery disease, type 2 diabetes, depression, prostate cancer, asthma, and so on and so forth. And then in each of the disorders of disease, and there are a triangle and a line. So what a triangle means is based on his clinical report, what is the disease risk, okay? So for example, for obesity, and his current clinical risk based on the cholesterol level, based on everything that's being shown, and it's about 27%. But his DNA is not doing him a favor, okay? So his DNA says his adjusted clinical risk is 60% based on his DNA sequence. And after all those database searches. And then for the coronary artery disease, and you can see this is 50%, and then there is adjusted disease risk. But some of them, so you can see that the arrow goes to this particular direction. That means although that based on his current clinical exam, and there is 10% of risk, but his DNA said your risk is actually smaller. So I guess, and what we can go a little bit more detailed for one of the lines. This is a myocardial infarction, and based on his current clinical risk, it's about 4%, and then with the disease, his DNA sequence adjustment, it goes to about 10%. So you can see if you click into this, and it also tells you how many SNPs are contribute to this particular decision, and then which SNP, how much effort, how much impact it is on. So that's why this is the initial risk, and this is end risk. Okay. I'm not saying I agree with everything that's being presented here in terms of the way that the different variants and how they are going to the adjustment. But I do see that this is a very, very interesting paper that in the future, in the clinical situation, that you go to the hospital, you examine yourself, and then it gives you, and then you have your DNA sequence that is always with you, and then you can readjust your clinical risk based on your clinical report. This is fundamentally different from the service like 23andMe and something like that. Because for those services, it's only based on your DNA sequence. Okay, it is only DNA, it defines who you are, but your clinical report tells us how you lived. Okay, in terms of our clinical risk, it's a combination of who you are and how you lived. So that's why I think using this type of analysis, for me, it makes more sense comparing to the ones that simply calculating the disease risk based on your DNA sequences. But that's my opinion. But they also go further, look at different the way that this person can metabolize drugs. This is more on the pharmacogenomic type of studies. So I'm not going to go detail. So for example, they will tell you this person is good to response to statin and things like that. And they also mentioned that different disease, they are actually linked together, and they are not coming to, I mean, if you got type 2 diabetes and obesity, and the chance you got coronary artery disease will probably be higher as well. So smoking is contributing to a lot of this. And they call it a systems biology approach to study the disease rates, but this is not too much associated with this one particular individual. Okay, so I think this is one of the first study of comprehensive analysis of human genome in a defined clinical context. In a defined clinical context is the key word. And I don't like to see only based on the genomic DNA sequence. I think it's overusing the ability of the sequencing technology and our knowledge. But you can read all this and the proof of concept analysis and the predisposition and risk to different diseases is interdependent. That is systems biology part of the story. The limitation for this type of study is a comprehensive database of rare mutation is needed. I think there are multiple databases on the market is trying to address this. And there are imperfections of all human genomes published to date, false positive, false negative. So there are always some of the issues. But I think the take-home message of this paper is one way to use the genetic information is not only to cure disease, but can be used in the regular clinical practice or even the annual check that by combining your DNA sequence with the clinical outcomes. Any questions so far? Okay. So the next section I want to talk about is more on the experimental considerations of the next generation of the DNA sequencing, and I will start from the whole genome sequencing. There's really not too much to talk about for the whole genome sequencing, and the number of sequenced genome grow fast in the cost of the sequencing jobs. And Watson genome in 2007 cost $2 million. And then there's 1,000 Genomes Project, there's TCGA, and many disease genomes are available. And they're all whole genome sequencing. And the cost now is about $1,000 per genome. And it's going down, hopefully going down in the future. But even with this number, in many cases, in some of cases, it's still cost prohibitive to study a disease. And because $1,000 per sample, it adds up pretty quickly. If you need 10,000 patients, and that is soon become a $10 million project, that is still expensive. So if we don't have enough resource to do the whole genome sequencing, one way to do that is to stay focused. So meaning that focus on the part of the genome that we know how to interpret their results. And a starter for that is most of people taken is if I don't have the resource to sequence the entire genome, I will just sequence the exome region part of it because those are the region that will become a protein. And if they have a variant that will change the amino acid sequence, the chances they are possibly to impact the phenotype. Okay, and so far that depends on different platforms to do the whole exome sequencing. The cost is about $350 to $600, and so it's very much becoming very much affordable for the whole exome sequencing. Okay. And there is also some of the studies, what they do is they are not targeting on the exome regions. Rather, they are only focused on a region of the genome that the people already know they are associated to certain disease. Because for many years, people already did linkage or GWAS study for some of the disease. And for example, one study that I'm involved that identified a region on chromosome 2, and we know exactly this region that is linked genetically to this particular disease. We just don't know which variant or which gene is impacting that. In this case, what we did is we will design the probes to target on those regions and only trying to sequence this particular region. So based on only exon regions or other regions that previously defined the associate to certain disease. All right, so saying that is easy, let's only sequence the exon. How do we get rid of the part that is not exon? Because they are going to be mixed inside the whole genome anyways. So here are the different strategies people have done to capture the region, the genomic region you're interested in. And that will include the long-range PCR, and I'm not going to go detail, it's possible, but and it's doable, and people are doing that for some of the disease, but this is by far not the most popular approach. And some people do the reduced representation libraries, and for example, using the cDNA libraries and to put down the regions so that you basically, you put down the exon regions because they have been transcribed into RNA, you convert that into cDNA, use that as a probe to target it. I mean, we're not using that anyways, so we're not going to talk too much detail. And then another one is to use the restriction enzymes. So basically, you use a pair or different combinations of restriction enzymes. They will cut at a specific part of the DNA, and then you use the size selection to select the ones that you're interested in. And we will talk about this a little bit more when we talk about the DNA methylation part. And there is a approach called the RRBS, which is a reduced library bisulfite sequencing, and to focus on the promoter and the enhancer regions to do the DNA methylation studies. But we will skip at this point. But most people are using is the capture array technology or capture in solutions. So this is the capture array technology. So if you look at it, this is the DNA and the genomic DNA. The first thing you do is you will do the DNA fragmentation, break it into smaller pieces. So now you are seeing some of the fragments are purple, others are black, and purple are the ones that you are interested in, or regions you're interested in. I mean, the issue is what's the best way to get rid of the black fragments? And the way you do that is that they put an adapter on it, the library, and then you are going to design a microarray that design the probes only target on the regions you are interested in. For example, if I'm interested in exon regions, I will design the probes to target on every single exon. So you can see they are sitting there. And then your DNA, purple DNA, because they are exon regions, they are going to be reverse complemented to the probe. So you can see they are being sticked to this array. And then the black ones are the ones that are not being, it's going to be not, I mean, it doesn't have anything to target on. And then at this stage, you wash everything else away, meaning the black part is being washed away, and you only have the purple part, and you sequence the purple part. So this is the exon array technology, and there are different players on that, and but most dominant ones are the Nimbogen, and now it's being purchased by Roche, and also the Agilent. They all have their own arrays. But now, because this array-based technology requires specific instrumentation to do that, and they are more or less being replaced by a very similar approach called capture in solution. And this is the figure that from the Agilent SureSelect assays, and which I will go through a little bit. But this one is targeting about 50 base of the exon regions, and they have some platform target on 71 base. And now they also have another one target on 45 base. So the way they do that is very, very similar to exon array. So you can see this is the genomic DNA, and you will first fragment the DNA into smaller parts, and then you mix your DNA with a bunch of probes that you designed. So you can see here is the DNA sequence you want to target, let's say exon sequencing, and these are the exon region, the genomic DNA sequence of those. And then you see there are a little little beads on that. Those are normally the biotin label. Okay, and then what you're going to do is you're mix all this together, and then your DNA is going to be reverse hybridized to the probes, and with this biotin being labeled there. So you can see that some of them that are being reverse complemented to the biotin side, others is not. And then what you do is you use this bead. This bead only capture those biotin signals, and then everything else will be washed away. And then now you only sequence this part that's being captured by these beads, and that's the way that we are able to capture the exon regions. And these oligos, it doesn't have to be the exon regions. It can be any region that you are interested in. And I think at least for adjuvant, you can just send in the region, genomic region you're interested in. They will automatically, they have a software to generate all the oligos, and they will send those oligos to you. Okay. So this is the capture in solution technology. Sometimes we think it's still too expensive, and because if you think about the cost associated to the capture, and there is, I mean, to do the whole exome sequencing or capture based, and the design the capture base, it costs some money, and the capture reaction itself costs money. If you have to do this one by one for every single sample, and this dollar is adding up, right? And then in addition to that, there will be costs associated to the sequencing, and will include the library preparation, which costs some money, and the sequencing. I mean, with the current cost of sequencing going so low, and this is actually $1 sign. And while capture reaction, the library prep, this is $2 sign here. Okay. And for exome sequencing, each sample still is expensive, and then sometimes that we are going to do the pooled sequencing. So let me show you how we are going to do the pooled sequencing. So for this, normally what we do is we got multiple people, their DNA, and in many cases, they will represent a similar phenotype. So let's say all these are controls, and there's another group of people, they are all patients or case. Okay, so what we do is we got their DNA, and then we will first mix the DNA, okay? And then after that, do the capture, do the library construction. Remember, for both these two steps, they cost money if you do those samples individually. And then if you do this way, and this will become one single reaction. So you put the DNA together, and then you do this. And for most of this part, you're initially you are reducing the cost by 8 times if you put 8 patient data together. Of course, the sequencing will still cost money, but this part will reduce the cost dramatically. Okay. In some of the cases, if you are building this based on an innovative statistical analysis, let's say if you have a couple of pools on the control, a couple of pools on the patient, and then by analyzing the data properly, you are still able to identify the genetic variants that are associated to certain disease. And this will help you to reduce the sample size. I mean, reduce the overall cost, but increase the sample size. Now you are bringing many more people, their DNA sequence into it. And so you identify genetic variants in a cohort of individuals, not only one individual. And you can identify the variants, but won't know which variants are from which individual. So this is one of the problem. But if your goal is to see which variant is associated to disease, in many cases you don't need to know which variant that individual come from. And but you also cannot target too big of a region. Normally, it's only less than 1 megabase of a region of your interest, and the analysis can be tricky, but it's certainly doable. And there are some of the studies that using similar type of approach and get very, very interesting results. Okay, so next I'm going to talk about the issues with short read sequencing for DNA sequencing. Okay, so far that every for the whole genome sequencing, for the whole exome sequencing, for the pooled sequencing, what we talk about is always using Illumina sequencing platform, which is a short sequence. What's the problem with that? So let's see if we have 2 variants in this particular individual. If our sequencing rates is only a part of it, okay? It's only cover a small proportion of it. So you can see in this region, we have A and G. In this region, we have C and TA, okay? And then our result is going to be in this region, we have A or G. In this region, we have, in this site, we have C or TA. That's the result we're going to get. But we will never know A and C should pair together. They're on the same allele, okay? And G and A, they are on the same allele. We will never know this. That make sense? But sometimes this information is important. So those are called haplotype. And if we use short read sequencing, we will not be able to recover this haplotype. So let's see what the solution for this. I mean, of course, you can use long-range sequencing like the PacBio or nanopore sequencers. They are sequencing very, very long sequencing rates. But as we mentioned earlier, that the accuracy of these technologies are not very good. And they have a huge amount of sequencing errors. So we will not trust the SNPs that are being caught by these technologies. And if we really want to call the SNPs, we still need to rely on short read sequencing like Illumina sequencing technology. And the way to do that is to use molecular barcode from the linked reads. And actually, we do have the instrument to do that. So let me tell you the way. So next slides, we'll have a schematic on how to do that. But if you look at this schematics, blue is from all the DNA fragments from one allele. And then orange is from a different allele, okay? And if we normally use a short read sequencing, we are going to mix them together to sequence it. We will not know which read is coming from blue, which read is coming from orange. We will not know that, okay? So in that case, if we do the assembly, we will kind of have to mix them up together. So the allele information is being lost. And if we have some way, and at this stage, we know which read is coming from the blue allele, which from orange allele, and then we can assemble 2 alleles separately, and then the haplotype information will be recovered. So one of the way to do that is to use the 10x genomics sequencing the instrument that we have in our center. So if you look at this, these are the bead, okay? And then this is the bead is going to be pushed towards this direction. And here is something called a high molecular weight genomic DNA and the enzyme. So those are the DNA. But this DNA is not a short 500 base DNA sequence. Rather, it's over 100,000 bases. Those are big DNA sequences. Okay, so each fragment is so that's why it's called a high molecular weight. So I think this thing died. That's why it's called a high molecular weight. So each of those DNA molecules, they are very, very long, 100,000 bases. And then you can see here is the DNA molecule will be attached to each individual bead. So we can control the reaction that way. And then after that, you are pushing this into an oil environment. Okay, so all these are water-based, and this is oil. So you can see that those DNA and those beads, they are being trapped within a bubble, right? So you can see every single one become a separate reaction units because they are not mixing together, okay? And on the bead, actually, there is a barcode for this specific DNA sequence. So by the end of the process, every single read that coming from this one DNA sequence will carry the exactly same barcode. And then the ones coming from this DNA sequence will carry a different barcode. And then by the end of the analysis, through analyzing the barcode information, we will know that which short-read DNA sequencing is coming from the same long high molecular weight DNA that initially being pushed in. That make sense? So basically, we will have okay, someone else was talking about. So again, let me lead through this again. Okay, so this is the DNA which is very, very long. Okay, and then they were being attached to a barcode, and then we'll push into this oil. So each of this bubble is a separated reaction units. And in this bead, and there are many barcodes. The barcode will carry the exactly same short oligo barcode sequences. So the reaction will occur within this, but the DNA will be further fragmented into smaller components, and those barcodes will be attached towards the end of the sequence. So all the DNA sequence that coming from the initial, the same long DNA will carry the exactly same barcode. Okay, so by that way, through analyzing the barcode information, we will know that which short reads are coming from the same DNA sequence. So if we do this properly, and then we will have the ability to identify something like this. So in this case, this is coming from the 10x website. So you can see they were able to identify two different alleles. And one of the alleles, there is 70,000 base of the deletion on this allele. You will otherwise not be able to identify. So they also developed some of the algorithms to do the allele-specific de novo assembly. And so far that I don't have the data yet, but from on Wednesday we are going to have the training. I'm not talking about this class. The Center for Medical Genomics will have the training and to do this experiment that we already get the high molecular weight DNA prepared, and then we will just go ahead to follow the protocol. So in 3 weeks we will have the data, and we'll see whether we can identify something like that as they claimed. So the last small proportion I want to talk about is the single-cell genomic sequencing. And this is different from the single-cell RNA sequencing that we will devote a full lecture on. And this is the single-cell DNA sequencing, which I will be honest, we don't have any experience on that. So I will just tell you the highlight information about a paper I read. So the challenge for doing the single-cell DNA sequencing is the efficient physical isolation of the individual cells. So those are all different challenges. Amplification of the genome of the single cell is very challenging, and the query of genome in a cost-effective manner to identify variants, and this is difficult, and interpret the data and in the context of bias and errors is difficult. But in general, that the single cell DNA sequencing includes several components. So you can see that starting from the sample or the tissue, and then the first part is you are going to separate the individual cell separately. Okay, there's several different ways you can do that. The first one is a laser capture microdissection, and this is very, very challenging and technically demanding as well. And you can use a fax sorting as well, and also you can use a microfluidic device. And I think in our institution we do have the C1 fluidime that can help us to do this. So I mean there are ways of all three type of instruments that I think our institution have it. So once you get to the single cell isolation level, you will need to, so basically for this DNA sequencing, my understanding is you will still push individual cells into individual tube. So if there's if you want to sequence 100 cells, and you are going to push them into 100 tubes, and then after that, you are going to treat each individual cell as a bulk sample, but keep in mind that they only have 2 copies of DNA in that, and then you need to do the DNA extraction, and using a very specific type of enzyme. It's called isothermal polymerase, and I have not used that, but it seems that they're keeping the fidelity very, very high. And then you prepare the library, and then you sequence in the library and doing the analysis. I think in terms of the difficulty part, there are 2 major parts. One is you need to separate the cells, and the second one is you need to faithfully amplify the DNA and with a very, very limited initial material to begin with. And there's multiple solutions for that. Again, this is an area I have no experience at all, and we have a lot of experiments on the single cell RNA sequencing, but for DNA sequencing, I don't know too much about that. Next lecture, we'll talk about mapability, refined alignment, and quality recalibration, and those are the latter 2 components are part of the GATK package. Okay, and any question? Okay, we'll see you on Friday.